Hey there, my name is Ahash and I am a 16-year-old genomics researcher at the Knowledge Society. Today I'm going to be talking about CRISPR's application to cardiovascular research. Let's check it out. So CRISPR has been an emerging field and an emerging application to many different diseases. Many different scientists and researchers have been trying to apply it into their diseases and their specialties. But what I found interesting among my different research was the cardiovascular approach. You may have heard of CRISPR's approach in many different other therapeutics. Just to name a few, we've been able to utilize CRISPR to modify our immune cells and make them have the ability to attack cancer cells via CAR T cell therapy. We've also been able to modify the genes of certain cells that are known to have genes that can cause cancer. And with cardiovascular disease, it's a different approach. We're not exactly targeting a specific mutation because of its ability to cause the disease. Rather, it's an indirect approach that requires preventative healthcare. And that's exactly how we can take the next step to save lives without them causing problems in the first place. So in coronary artery disease, also known as CHD or CAD, what happens is that the coronary arteries that supply blood to the heart that carry important nutrients, get clogged up. Now this can be a result of low density lipoproteins known as LDL cholesterol, glucose or other types of plaque depositing agents. Now, the coronary arteries are very crucial to our body because it supplies blood to the heart. If we don't get blood to the heart, then the heart stops. And so the rest of our body doesn't get blood from the heart, and that can cause severe damage, resulting in myocardial infarction, also known as a heart attack. Now, atherosclerosis, the process of forming plaques, can be reduced, and this is often encouraged by leading a healthy lifestyle and reducing factors such as high glucose levels or cholesterol levels. But in all attempts, often this results in it being the #1 killer among the world, heart attacks. Scientists have been researching many different ways to actually produce a lower amount of patients dying from this disease. But it's been very difficult provided that it can happen suddenly and the heart is very silent. But let's talk about a more specific issue here, and that is LDL cholesterol. Now, LDL cholesterol is low density lipoproteins. Now, this is one that I spoke about a bit earlier, but this type of cholesterol is bad because it can actually produce bad results such as coronary artery disease, whereas the better alternative, which is high density lipoproteins, actually reduce the amount of plaques that form. One of the main targets that has been known to reduce LDL cholesterol recently has been known as a PCSK9 gene. Now, the PCSK9 gene also hosts proteins which are LDLR receptor antagonists. So let's break down a bit. LDR receptors, what are they? Well, LDLR receptors are essentially receptors for the LDL cholesterol. Now, what they do is basically receive LDL cholesterol, and often receptors tend to transfer it into a cell. So if it's not in the cell and it's not being used, or it's not even in any type of cell, it can result in damaging outcomes when it's just laying in the bloodstream. So these are where LDL receptors, can come into play. Now, when we're looking at LDLR antagonists, just like in the movies, antagonists tend to be bad because they actually block the function of this type of cell or this type of receptor. So when we're looking at the PCSK9 gene, provided that it is an antagonist, we can find that if you have a gain of function mutation and an overexpression, what happens is that you tend to actually have more LDL cholesterol laying around in your bloodstream. Now that can be dangerous. That's where we want to target. Okay, but what are we going to do now that we know that PCSK9 can actually be an antagonist? That's exactly where CRISPR-Cas9 can come into play. As soon as we found out that PCSK9 can be an antagonist, many different pharmaceutical companies and scientists have been working on developing antibodies. However, because they're antibodies and because they're drugs, ultimately they start to wear out and you kind of need a refill. But with CRISPR-Cas9, we can actually permanently alter this and make sure that hepatocytes don't release PCSK9 more often. And that's exactly one way to preventively tackle coronary heart disease. But how's it gonna work? Let's get into it. So this particular type of protein was found in hypercholesterolemia patients, especially autosomal dominance. So in this case, what happens is that they actually had to analyze this, and they tested this hypothesis of modifying the PCSK9 gene with rats. And so they tested two things: adeno-associated viruses and also the efficiency of the CRISPR-Cas system. So in order for this to happen, what they did was that they first designed a guide RNA. Now this particular guide RNA basically has the ability to bind to the target DNA site, and the CRISPR-Cas system can lead the Cas9, the endonuclease, to the target DNA strand. Another thing that then binds is also known as the protospacer adjacent motif. This basically can differentiate the target DNA sequence and the guide RNA, and that way the Cas9 has the knowledge in terms of where does it want to cut. Now, once this cut is performed on the target DNA strand, it forms a break, a double-strand break, which can be filled in with the donor template via homology-directed repair, which, on the contrary to NHEDG, non-homologous end joining, actually is more efficient because it reduces the amount of off-target mutations, and also you can insert your own DNA template. Moreover, you had to encapsulate this RNA candidate that performs all the magic within a adeno-associated viruses with different expressions, for example, co-expression Cas9. But they also tested this with a plasmid. And so when we're looking at this, we can see that adeno-associated viruses can be used for viral vectors, whereas it can also be compared to plasmids. And they found that it could be have the potential to not be as efficient in terms of the comparison between plasmids and adenoviruses. However, it's still under research and study. But what they did find was that LDL cholesterol levels were lower. On the contrary, HDL receptors and HDL levels in general were not changed. So that means that it is right now successful, and this poses a whole new specific type of treatment for cardiovascular disease. So ultimately, this is a really cool application for CRISPR research. I hope you enjoyed that video. This is Ahash signing out.